Hi, everyone. Welcome. Today we have as our presenter Jorge Vera Buschmann. Jorge was born in Chile and received his PhD from University of Chile in neuroscience and was mentored by Magdalena Sanhueza. Then he received the Grass Fellowship while he was in Chile and subsequently the Best Thesis Award in 2017. He then came to the United States, joined Kamran Khodakha's lab at Albert Einstein University in New York, where he is now an associate researcher. He recently won the Best Poster Award at the Dopamine Conference, which was held in Montreal because of the work that he's been doing with his wife on understanding the link between the cerebellum and the substantia nigra. Jorge, thank you so much for being here.
Thank you, Reza, for first organizing these wonderful seminars and then for inviting me. I have to say here that I'm representing here the Kodaka lab, and the idea is to show the work that we do here in the lab and that we have been doing in the last 7 years or so since I joined the lab, related to the communication of the cerebellum with the dopamine system. So first, I would like to start with the main role that the cerebellum does. That we think we agree that it optimizes the behavior by doing predictions, right? So it does this prediction because it's a neuronal prediction machine that do association of sensory motor activity, and we use those predictions the whole time. Normally, we are not aware of that predictions. I like to use the example of the coffee cup. For example, every time we grab something, we have to do an estimation of the weight to then do the appropriate muscle action in order to do a smooth behavior when grabbing the cup. And that is just one example of all the different predictions that we are doing the whole time, and that we have really good evidence that the cerebellum take part on it. Other way to see this prediction is seeing them as a way that the body, in particular the cerebellum, learns the causal structure of the interaction of the animals with the environment that are vital to do an optimized behavior. So we have the good information, and all this idea comes from the study related to motor behavior, but we also have a good amount of suggestive evidence to propose that these predictions are also used for cognition or for non-motor behaviors, suggesting that the cerebellum, with this ability of predicting, is contributing to behaviors beyond just the motor domain. And for we have a lot of information, and we know a lot about how these predictions are built inside the cerebellar architecture. We know that the cerebellar cortex receive input from all the sensory modalities, and they process and do this association, and then sends out to the rest of the brain through the deep cerebellar nuclei, which is like the main output of the cerebellum. But we know very little about how this information, these predictive signals that are encoded by the cerebellum, reach the downstream structures in the forebrain. The main idea that we have is that this, based on a really good evidence, is that cerebellar signals go to the rest of the brain through the thalamic relay using glutamatergic signals. But now, thanks to the development of new tools for anatomy and physiology, we are starting to learn that there are more pathways beyond the thalamic glutamatergic one. And what I want to present today is the work we do here in the lab, characterizing how the cerebellum contacts the dopamine system. And by doing this, it converts or implements these prediction signals in the form of dopamine, or it may actually. We are working for that. So the dopamine system is a very essential system for behavior. Dopamine, the molecule, is vital for movement, for reward, and for learning. We know a lot about the involvement of dopamine in movement due to Parkinson's disease, where the circuit that provides dopamine to the region involved in control of movement degenerates. Regarding the involvement of dopamine in reward processing, we also know a lot because of the involvement in mental disorders like schizophrenia, autism, or addiction. And they were initially described as working or encoding the detection or prediction of reward. And at this point, despite that we know a lot, this field, for the same reason of having new tools to investigate the mechanism by which dopamine works, is a field of intense research. So by doing this work, we are trying to mix cerebellum field with the dopamine field. There is substantial evidence linking the cerebellum to the dopamine system in general. The literature is vast. There is plenty of anatomical, functional data in humans, in healthy humans and patients with mental disorders, animal models linking cerebellar function and dysfunction to the dopamine system. But now, the first direct confirmation and functional evaluation of this connection from the cerebellum to the dopamine system came with a paper from the lab in 2019, where we described direct projections to the dopamine neurons located in the ventral tegmental area that are related to reward processing. Now, regarding the movement part, we just published a paper in which we do the first evaluation, the direct confirmation and evaluation of the direct projections from the cerebellum to the neurons located in the substantia nigra pars compacta that are involved. These are the ones that degenerate in Parkinson's and are involved in movement. So with these two papers, we basically are providing the foundation of this research that try to address the connection between the cerebellum and the dopamine system. So a brief anatomy and function of the dopamine system that we are going to use along the presentation. As I commented before, in this talk, we're going to focus in three main dopamine pathways. The first one will be the one composed by the neurons located in the substantia nigra pars compacta, or SNC, that provides dopamine to the striatum, where these dopamine signals are involved in setting the kinematics of movement, the initiation, and also the speed. We also will see in the other half of the presentation the pathway conformed by the dopamine neurons located in the ventral tegmental area that provides dopamine to the nucleus accumbens, where they are related to reward processing in general. That is required for goal-directed behavior. And also, these neurons, an independent cell type, provides dopamine to the prefrontal cortex, particularly important for this talk, to the medial prefrontal cortex that is involved in decision-making and behavioral flexibility, and is supposed to require dopamine for these processes. So, yeah, this is in the opposite way that I'm gonna do it in the presentation. I'm gonna be talking about the follow-up of the paper done by Ilaria Carta and Christopher Chen with the analysis and validation of the anatomy and function of the circuits that describe the cerebellar input and its influence to nucleus accumbens and prefrontal cortex. And also, I will summarize the most important observations that we did in the characterization of the inputs to the SNC, and the role of the circuit in movement, with some with one unpublished data that is a follow-up of this observation. So as an outline, this is how the circuit looks in the mouse model that we used. Something that I forget to mention that is relevant for this presentation is that we have done all this characterization in mice model, but it has been described in two papers that these projections from cerebellum to SNC and BTA are conserved in humans. So our observation should apply or could be extended to us. Okay, so starting with the SNC circuit, how we call it for short, I'm gonna first show an anatomical characterization, then functional evaluation of the circuit, and then potential behavioral role. So first thing that we wanted to do was to see where are located in the SNC the neurons that receive projection from the DCN. For that, we injected anterograde tracers that will label the postsynaptic cells, and we mapped them. So we could identify in the SNC the neurons directly by the GFP expression, and by staining against TH, we could identify them as dopaminergic and non-dopaminergic. And by doing this, we could map them. We found that neurons along the whole anterior-posterior axis of the SNC receive input from the DCN, showing first that these inputs are, or this circuit is massive, it's not small. And second, that it is known that different regions of the SNC project to different regions of the striatum. Thus, having these neurons in the whole extent of the SNC suggests that the cerebellum is contributing to or participating to this dopamine system in at its whole extent. So as you can see here, to do this labeling, we use as a starting point the 3DCN. So we don't know which DCN is producing this projection. So in order, and it is important to know which DCN is contributing to this circuit, because we know that different DCN have different roles. So by knowing which one contributes to this circuit, we might have an idea of potential function of the circuit. So to do that, we use a retrograde tracing, non-selective, in injecting in the SNC, and we found positive cells in the 3 deep cerebral nuclei, showing that, as expected, the cerebellum is using all its functions to provide signals to the SNC. Now, we have the caveat here that this tracer is not synaptic specific, and it doesn't label specifically projections to dopaminergic neurons. So in order to do that, we use a rabies tracer, mapping specifically the neurons that project to the dopaminergic neurons, and we found similar results. Basically, that neurons located in the 3DCN provide inputs to the dopaminergic neurons in the SNC. Okay, so now we're gonna assess the functionality of this circuit. One thing is to have the circuit and the connection, but other thing is what happens with the function. So this first thing we evaluated was the capacity of cerebellar inputs to activate dopaminergic neurons. To do that, we expressed cholinergic in the DCN and excited cerebellar axons in the SNC at the same time of recording with an optrode the neurons from the SNC, and we found that after activating cerebellar axons, SNC neurons respond rapidly with a latency of a couple of milliseconds, showing that activation of cerebellar inputs can drive efficiently the SNC neurons. And more importantly, when applying a train of stimulus at 20 hertz, in this case, SNC neurons can reliably follow the pattern of activation of cerebellar inputs. And this is important because we know that the DCN cells act by firing at in trains of action potential based on sustained activity that they have. So this means that SNC neurons have the ability to follow the fluctuation in the activity of DCN neurons. We know, it is known that SNC neurons provide with dopamine to the striatum. So we wanted to see whether cerebellar inputs were capable of contributing to dopamine signals there. And for that, we did a similar approach, activating cerebellar inputs to SNC with optogenetics by monitoring dopamine signals with delight sensor and fiber photometry. When doing this, when we saw that just a brief activation of cerebellar axons with 1 millisecond pulse was enough to see a clear increase in dopamine in the striatum. And more importantly, when we mimic DCN input with a train, we see that there is first a phasic response that then gives rise to a sustained increase in dopamine that lasts the duration of the cerebellar activation, suggesting that again, dopamine neurons are providing dopamine to the striatum, following the pattern of activity of cerebellar inputs. This, as I commented in the introduction, this circuit is known to be related to movement, the SNC to the striatum. So we wanted to see whether the cerebellum was participating during movement. For that, we express a calcium sensor to monitor calcium activity in cerebellar axons in the SNC, and to see at the same time the activity of dopamine neurons, we expressed a red calcium sensor. So through the same optic fiber, we could monitor the activity of both components of the circuit. And we saw that when mouse engaged in spontaneous locomotion, there was a co-activation of both cerebellar axons and SNC neurons every time that the mouse was walking, suggesting that cerebellum indeed is participating in movement. So to have a more causal approach to test the contribution of cerebellum to movement, we did a similar experiment, but in this case, we wanted to activate cerebellar axons and see what was the response in mouse walking. So for that, we again expressed channel rhodopsin to activate cerebellar inputs with optogenetics. And we did 3 types of stimulation, starting with a small single pulse and finishing with a long 3-second train to produce an increased amount of cerebellar activation. Please note that here I'm doing this pattern of activation during a block of 5 minutes, where I give the stimulation and then I wait for 5 to 10 seconds. So there is like a pattern of activation of the cerebellar inputs. When doing this, and as expected from the previous slide, we see a proportional release of dopamine in the striatum, showing that the more stimulation, the more dopamine in the striatum. Interestingly, we found a proportional effect in the speed of walking of mice when doing this cerebellar activation. So the more stimulation, we see the more walking and faster movement. To see more closer the relationship between the effect in walking speed and dopamine, we plot together these 2 variables, and we saw this very strong correlation, suggesting that the more stimulation of the cerebellum, the more striatal dopamine, and then the more speed in the walking of mice. So at this point, we didn't have, this is what is published in the paper, we didn't have a clear idea of how the cerebellum was producing this increasing movement, but given this correlation, it suggests that it might be doing it through an increase in striatal dopamine. So in order to test that possibility, we went to do a second manipulation. So the idea was to activate cerebellar inputs to promote movement, and at the same time to inhibit a dopaminergic neuron in the SNC to get rid of the striatal dopamine and see if that could prevent the effect of cerebellar stimulation. Indeed, that is what we found. So when repeating our stimulation to increase the movement of mice, we see the increase in speed, reproducing our previous observation. And then when silencing SNC dopaminergic neurons, that should produce a reduction in striatal dopamine, we see a decrease in the speed of mice coming back to baseline levels and making cerebellar activation no longer active in promoting the increase in speed, thus supporting the idea that the way that the cerebellum is contributing to this increase in speed is by increasing striatal dopamine. Okay, so now switching gears to the non-motor domain of this circuit and how it is related to reward processing. I'm going to show like a key experiment where we tested whether cerebellar inputs have a proportional activity to the amount of reward that the mice are processing. To do that, we train mice in a Pavlovian reward association task, and within the same session, we give them two different rewards, like a normal normal water with like a low level of value, and also sweet water as a stimulus with high reward value. And what we saw is that cerebellar, the activity of cerebellar axons contacting the SNC was higher when processing the high value reward. This is an observation that is similar to what happens in the SNC neurons that are known to encode reward value by an increase in the level of activity, thus suggesting that cerebellar inputs to the SNC contain reward value. So we in the paper, that is where when we end the paper, having the characterization that cerebellar inputs contribute to set the speed of walking, but we have the limitation there that mice were not in the context of reward processing, and also mice were head restrained. So it is not the better approach to test the involvement of the circuit in control of movement and reward. So to have these two dimensions combined in order to assess the role of cerebellar inputs in the striatal dopamine, we are implementing this task where mice are trained to run at different speeds, and they learn very well that, and they run at different speed in order to get a reward. So we can by training them, we can control the speed at which they run. So we can control whether they run faster or slower at the same time that we are measuring, and also we can manipulate the cerebellar inputs to the SNC. So we expect that with this task, we can study deeper the contribution of cerebellum to movement speed and also to reward processing through this pathway. So as a summary of this part, I just showed you that the cerebellum established direct projections with the SNC, that via this projection, it contributes to dopamine levels in the striatum, and that these cerebellar inputs to the SNC convey both motor and reward-related information. And it remains to be determined the precise role of these signals in the context of movement, speed, and reward processing. And that is what we are doing with this last task that I showed. So now moving to the other half of the presentation. This is all unpublished and is the characterization of this pathway connecting the cerebellum with nucleus accumbens and prefrontal cortex via the VTA. I'm going to follow like a similar structure of the presentation. So first, we did the mapping similar to what I just showed you for SNC, but then we want to quantify the neurons in the VTA. We can identify them as dopaminergic or non-dopaminergic, and we found, similar to SNC, that these postsynaptic cells to DCN are spread all over the way, over the place in the VTA, and knowing that the different subregions of the VTA provides dopamine to different downstream regions. Among those, nucleus accumbens and MPFC, this data suggests that the cerebellum is contributing to dopamine signals to basically everywhere. So to test that possibility and to see the pattern of projection of these DCN recipient cells into the nucleus accumbens and MPFC, we did a similar experiment, but here we complemented the VTA neurons with a Cre-dependent fluorophore in order to label those cells and then map those axons. So by doing that, we can have access to the axons of the VTA neurons that receive input from the DCN. And when we look in the nucleus accumbens, we see these axons that most of them are dopaminergic, spread all over the place in nucleus accumbens. Similarly with the MPFC. So we can track these the axons of dopaminergic neurons that are in the VTA that receive input from the DCN, and they are all over the place in the MPFC too. So next, what we did, similar to the SNC, is to see which DCN nuclei are contributing to this pathway. And for that, we now, since these circuits are dysynaptic, we have to go with more detail in the tools that we use. And here we are using a Radis type that allows the mapping of the VTA neurons that projects to the MPFC. So by tracing back these neurons, so VTA neurons that project to MPFC, then we can look for positive cells in the DCN. When we do that, we see like beautiful labeling of DCN cell in, again, the 3 DCN nuclei. This is the mapping that shows that the 3 DCN nuclei provides inputs to the VTA that then goes to the MPFC, suggesting again that the cerebellum, by all its modalities, may be contributing to signals to the MPFC. We did a similar approach with the nucleus accumbens, and to don't bother you with very similar results, I'm just going to summarize saying that with the projections to the nucleus accumbens via the VTA, we also see DCN cell labels in the 3 nuclei. Okay, so now the function. This is data extracted from the science paper where they show that using similar approach of the optogenetic activation of cerebellar axons in the VTA cells also respond very fastly with single pulse and also with trains, similar to what we saw in the SNC. VTA neurons are capable of following DCN activity, or this data suggests that. So then, first we wanted to see whether DCN inputs to VTA was capable of contributing to dopamine in nucleus accumbens using fiber photometry and dopamine sensor. And similar to what we saw in the dorsal striatum, here in nucleus accumbens, a single pulse is highly effective to produce dopamine signals in nucleus accumbens, and the train that simulates DCN inputs produce this sustained increase in dopamine that lasts the length of the stimulation. So we know that cerebellum is driving VTA cells. This is producing dopamine in the striatum. But what about the activity of nucleus accumbens, which are the ones that encode the reward? So to investigate that, we did acute recordings of single unit activity in nucleus accumbens, and we found that after the stimulation of cerebellar inputs to the VTA with a brief pulse, we saw like this increase in activity. that lasted a couple of 100 milliseconds and has like a dominant response peaking at about 200 milliseconds that agrees with being produced by the dopamine that is released and that has like similar kinetics, showing that cerebellar inputs is not only providing dopamine, but also modifying the activity of the neurons that are encoding reward and guiding goal-directed behavior. So what about the function of this branch of the circuit during reward processing? This is a similar experiment, the evaluation of cerebellar inputs under the consumption of reward with different value. And similarly, here after and this is inside the same session, when mice are consuming a higher reward, or a reward with higher value, they show a higher activity in this circuit, suggesting that these inputs participate in reward processing. So the other circuit that is involved here through the VTA, particularly for me, is the most important one, is the medial prefrontal cortex. So now I'm going to show you what we found there. What about my MPFC? So here, first regarding dopamine release, and I have to tell you that might not be interesting for or very exciting for the cerebellar field, but it has been difficult to track dopamine signals in the MPFC. So when I went to do this experiment to measure the effect, whether cerebellum could contribute to dopamine signal in MPFC, I saw this massive increase in dopamine that interestingly accumulates over time slowly during the length of the train. And you mentioned my humble poster award in the dopamine meeting. I'm pretty sure that it was because of these recordings in MPFC, because people love them, showing that cerebellar inputs are highly efficient increasing dopamine. So here we went a little further, confirming that we are engaging the dopamine neurons to produce this release. So we did an optogenetic and dread silencing of dopaminergic neurons selectively in that mice. And when we silence or when we inhibit dopamine neurons while stimulating cerebellar inputs, we see a drop in the release of dopamine to more than 50%, supporting the idea that with this manipulation, we are directly activating the sub-bodies of dopamine neurons and then releasing dopamine in the medial prefrontal cortex. As I commented when describing this dopamine kinetics, I want to highlight the point of how different the kinetic is compared with what we see in nucleus accumbens and the striatum. And here, I have to acknowledge that we are in a privileged position, that we are evaluating the dopamine system from outside, and we can compare the kinetics in these different regions. So this is how it looks, the dopamine evoked in MPFC with a 3-second train. And this is how it looks at the same scale in nucleus accumbens, that is very similar to what you find in the striatum, showing that particularly for the interest for our interest, that cerebellar inputs driving the VTA neurons will produce complete different effects in terms of dopamine signals in the MPFC than in the nucleus accumbens on the striatum, and this should be related to the function of the different circuits in behavior, and to have an idea of the dramatic effect that this different kinetics has at the moment of the integration of signals. When you do a sequence of trains of activation of cerebellar inputs that could mimic a consecutive processing of reward, for example, the way we see in our task, we found that the dopamine signals in MPFC accumulates over time and produce like a high increase of the baseline levels that last for seconds or minutes, and that doesn't happen in the nucleus accumbens or the MPFC or the striatum. And we think that should be important for the processes that happens in the medial prefrontal cortex, decision making and behavioral flexibility. Okay, But we know that the cerebellum works at a fast time scale and it's like a machine good for precision. So what happens with fast signals in the MPFC? For that, we did electrophysiological recordings in MPFC all over the place in MPFC. And we were very surprised to see that the main representative response in MPFC is a fast activation of neurons that occurs in about 80% of them. Here I recorded many mice in all different conditions, and this is the big summary. I'm talking about the 1,000 units, and this is highly reliable. And this fast response happens with a peak occurring at about 50 milliseconds. So together with having these slow dopamine signals that build up over time, we have these fast responses in MPFC, showing like a tight coupling in the function of the cerebellum and MPFC through the VTA. But we know that here there is a caveat, because it is possible that when activating the VTA, we might either producing unspecific activation of other regions, non the VTA, or maybe activating collaterals that go to the thalamus. So in order to confirm that we require the VTA to convey this activity to the MPFC, we did two sets of experiments. First, we did pharmacology, blocking the fast glutamatergic transmission in the VTA by infusing a CNQX. And when we do that, we found that the fast responses in MPFC are gone, so suggesting that they require the activation of VTA by glutamate input that should be provided by the cerebellum. And knowing that pharmacology is not the most clean approach, we also did optogenetics, where we can directly and selectively inhibit the VTA neurons that are located below the fiber where we are activating the axons. When doing this, when we just do the optogenetic activation, we see the increase in activity that I just showed you. And when we do this activation in the background of the silencing of VTA neurons, we get rid of this evoked response, further supporting the idea that when we do this manipulation, we're actually activating the VTA neurons that then convey this signal in terms of dopamine and excitation to the MPFC, and that should be required for cognitive processes. Okay, so now reaching the end of the talk. This is mostly a functional, anatomical and functional characterization, and doesn't tell much about whether these cerebral signals really have a role during behavior. And for that, we need a behavior that exploits the cognitive aspects in which dopamine is involved. And for that, as a future direction, I show here that we already implemented a cognitive task that exploits this. Here, mice learn to follow a rewarding rule, moving the wheel to the left or to the right. This task is a modification we did here in the lab from the one published by the IPL. And mice need to switch the rewarding rule from the left or to the right, exploiting behavioral flexibility that is known to require dopamine. So what we are doing now is to record or to monitor and manipulate cerebellar inputs to the VTA while mice are performing this cognitive task. And at the same time, we are monitoring the motor component as much as possible to then distinguish or correlate cerebellar activity with motor component and non-motor component, and see whether or where the cerebellar signals are playing a role during this cognitive task. And with that, I would like to finish with this summary. And this nice scheme that we use is a modification of the one we use in the review we have in Journal of Neuroscience, that the cerebellum established monosynaptic connection with the main dopamine center of the brain, and particularly with the SNC and the VTA. We haven't tested the other ones, but I can bet you that it also projects to the other dopamine centers. That via this connection, the cerebellum modulates dopamine in the striatum, nucleus accumbens, and prefrontal cortex. and that cerebellar signals to this dopamine center seems to convey both motor and reward and non-motor information, and that the precise properties of these signals, the behavioral role, remains to be determined, and that we're actively working in having this answer, and hopefully we're going to have them soon. Final part, one of the most important, the acknowledgement, thanks to Cameron Kodaka for being like the greatest mentor. He's the big mind behind all what I showed you. Throughout his career, he developed this research field that I will be proud of continuing then later when having my own laboratory. The lab members that participated in this work that I showed you, particularly, here, the lab, this is the current lab member, this is former lab members, particular mention for Maritza Oñate, which is my wife, and she is the anatomist here in the lab, and she did all the anatomy that I showed you. Also, a special mention for the rewarding group, Junichi Yoshida, Leila Hatami, and Farsan, that they did the experiment related to reward value assessment in both SNC and the VTA. And at the end, of course, thank you very much for the people that help us to experiment, and to our valuable collaborators that help us to do this beautiful experiment that I just showed you. And to the end, the nicest picture of the Kodaka lab having fun. And with that, I finished. Past 3 minutes, I think it's fine.
Thank you so much. Jorge, that was absolutely wonderful. Wonderful. So let's spend some time asking questions from Jorge. It was beautiful work. So I'm going to begin with a question. I know Farzana has a question for you as well. So the neurons that project from the deep nucleus to the SNC and VTA, as you showed, they are in all 3 nuclei. Do they only go to the dopaminergic cells, or do they go to other places in the brain or other inner neurons in those regions?
Yeah. No, we know that definitely there are collaterals sending, so sharing this projection to other brain regions. Until now, we haven't done a deep analysis of that because we've been busy characterizing mainly the projection to these regions, but we do know that there are collaterals to the thalamus, for example. And also there is some degree of collaterals sharing information between the VTA and the SNC. The amount of that and how functional or relevant for these experiments we do we don't know. But they do send collaterals. Okay.
Farzana, you had a question.
Hi, and thank you, Jorge. Yeah, so thanks for your talk. So yeah, I had a question. Those fast inputs to medial prefrontal cortex, are they glutamatergic?
We haven't tested. And here you open the Pandora's box that I was waiting for it, because it's the more exciting part of the work, is that I didn't mention that the VTA neurons are very special neurons. So in the VTA, you have dopaminergic and non-dopaminergic neurons. From the non-dopaminergic, you have glutamatergic neurons that project on PFC. So there is a predominant glutamatergic only projection to the MPFC that might be driving this fast excitation. You also have co-release of glutamate from dopaminergic neurons. So there could be also glutamate co-released. And the last possibility is that during the last 5 years, Pascal Kaiser has been promoting the idea of fast dopamine release in the dopamine system, not using this canonical volume transmission, but having dedicated a specialized release site that produce dopamine increases at the scale of milliseconds. So all the options are open, and we've been dealing a lot confirming that the activity that we see goes through the VTA. And I didn't mention, but we also know that it doesn't go through the thalamus, because if you inactivate the thalamus, you still see this beautiful evoked response in MPFC. So there's where we are. So I don't have a clear answer, but there are clear 3 options. For
that was very clear and helpful.
Yeah.
I have a quick technical question also. In a lot of your diagrams, you showed 475 something for excitation, for activation and record. So those are pretty close, but how did you deal with the spectral overlap? Was it like you did an image during stimulation?
When we do 2-color or 3-color experiments, we use the photometry system from neurophotometrics that do interleave excitation. So at any time, we only use one spectrum. So that's why we can split the signals. Yeah.
Thank you.
No problem.
Wonderful. Any more questions for Jorge?
I just one quick technical question, Jorge. Wonderful talk.
Nice seeing you.
Hey, how are you doing? Yeah, good to see you. Wonderful talk. I really enjoyed. Just one quick technical question. I just noticed that the dopamine signal that evoked the signal that we recorded in different regions, from compacta to last to striatum.
Ramki, I can't understand. I don't know if there's the audio or.
Can you hear me well?
I can hear you, but little like not the best way. Yeah.
I'm in the lab running the experiment.
Yeah, sorry, acoustic is not the best. Yeah.
Yeah. So the question is just a simple technical question. When you use the train of stimulation, I believe you use 20 years stimulation and you recorded the dopamine signal. I just wonder, I noticed that the signal, initially the amplitude of the signal shoot up and then goes back to certain level and then the space maintains the space.
I got it.
So whereas when you use the TTL pulse, in one case, it goes like sort of like up and down. The signal, which in a way gives an impression that it's somewhat following the train of your stimulation. I'm just wondering why you get this kind of different kind of evoked response.
The dopamine, but are you trying to compare the dopamine and the electrophysiological responses? No?
It's both, I believe it's both the dopamine signal. One dopamine signal where you recorded in the prefrontal cortex to your TTL pulse. Whereas in other cases, we use the orthogenetic stimulation, if I'm not wrong, with the 20-year stimulation, train of stimulation.
I don't understand the question. Sorry, Ram. Sorry.
You measure the
audio, right? Your audio is.
I'm sorry.
Yeah.
Your audio
is good. Okay.
It's the audio. Yeah.
Maybe I'm sorry about it.
So
Never mind.
It's fine, but maybe Reza, someone in your lab got the idea, but.
No, we did not. Maybe you want to type your question.
Oh, that is a good idea. Ramki, type it. Or
all right. Yeah. All right.
Let's see if there are any other questions for Jorge while Ramki types his question.
Yeah.
Jorge, the increased activity.
Okay, never mind. I'll catch up with you later because I'm wearing the gloves now. It's not very handy to type. I'm sorry about it.
I'm sorry. I didn't get it, but I bet you it was like a very interesting question.
Not very handy. That's fine.
Sorry. Okay. Thank you for jumping in, though. Reza.
So the increase in dopamine around movement onset is something that has been reported now for about, 10 years or so. And and of course, in the cerebellum, you do expect to see changes in activity when a movement takes place and so DCN definitely would show modulation with the movement. Typically on both sides of the cerebellum, this will happen bilaterally, potentially depending on the kind of movement that's being made. Do you see any laterality in the inputs to the SNC?
Oh, I mean, regarding what you just said, I could extend in 20 minutes. But regarding specifically that question, we see that the projections, and I didn't mention in details, but they are bilateral, but mostly with a bias to be contralateral regarding the anatomy. In terms of the function, when we see reward movement related, or let's say reward related signal, they are bilateral and identical in both regions. So in that sense, we don't see laterality.
Okay. Any other questions for Jorge?
If someone in the future, this video should go to YouTube. If someone in the future see this and has a question, please shoot me an email. I'll be happy to answer.
Wonderful. Wonderful. Okay. Thank you, everyone. Great to have you.
Thank you for the invitation. Bye-bye.